Well, my connection with our speaker is kind of a roundabout. I had been working with a
Oops.
project with CSX Railroad, and I was talking to Jason Paul, and I said, one of the things that we were interested in is that there's a lot of railroad ties along the river that are spent railroad ties, and uh, the whole river is priority habitat under natural heritage, and we'd like to have those picked up. But I've been working with him to get that done. And as we're having a conversation, he says, "Oh, you remember Trout Unlimited?" He says, "My brother Tim does a lot of like research and stuff, and he's so into like habitat restoration." And I said, "Do you think that he would be willing to speak?" And I'm like, he said, "Sure." So I called him to talk about railroad stuff, and he was able to contact his brother Tim, our speaker, and we had a nice conversation. And we kind of explained what we were doing here in our chapter, and we talked about what he has been involved in his career, and sent me a description. Uh, so Paul, our Tim Paul's our speaker. Bio after spending his youth in the creeks and ponds of farmlands of Indiana, he attended college in Illinois. Paul took a job working with wild salmon in the wilds of Alaska and has been wearing waders and working on aquatic environments ever since. He currently resides in Wisconsin, working for the U.S. Forest Service National Research Station, the Northern Research Station. His career has provided him with the opportunity to work in a variety of ecosystems, from high mountain deserts to temperate rainforests. He's been involved with a wide array of projects, including passage and enhancement, adequate aquatic habitat protection and restoration, population studies, landscape genetics, and even has dabbled in finfish aquaculture. And
Good. Thanks everybody for having me here. I got a couple of things I want to comment on briefly about your meeting there. First of all, seems like you guys are very active and doing a lot of great work there. So kudos to you all on that. And I know it takes a lot of effort and drive to make things like that happen. So that's exciting to hear that that's happening out there. I also wanted to point out, I saw some extra tops on somebody, and whoever they are probably knows who they are. They're the brown boots. You might see a few of those pictures or me wearing them here coming up. You're doing pit tagging, riparian work, habitat, in-stream work. You've got a trout DNA study going on, a brook trout resiliency study, and doing a lot of education. Those are the things I gleaned just from that 30 minutes there. And some of those are going to tie in directly with what I've got to say, which is pretty exciting. I won't promise that there's collaboration, but I think when we get to that point, I'll talk a little about what's going on with me. I wanted to kind of start with we'll get to where did that go? Let's move that. Can I? Yeah, let's move this up here and then we'll move. All right, let's try this. I just wanted to run out a brief biography of myself just so you'd see where I was coming from. And Randy covered some of this, so I won't hit it hard. But grew up in Indiana, so flatlander by youth, and then went to school in Illinois. I got a BS in environmental biology, and pretty much did a couple internships. One was with the wetland delineation, and the other was here on. There we go. I got to
Yep.
be on that screen to drive my. Okay, because I want to look at you guys and not at this, but we're getting there. Technical difficulties on this one. Sorry, guys. Or and everyone. So I did the wetland delineation thing in Kansas while I was in school, and then between my junior and senior year, I ended up in Southeast Alaska on an internship with the U.S. Forest Service, and kind of got me used to the West. I had really not traveled much out west and been around mountains, oceans, a variety of wildlife I hadn't experienced. And so, but that was fun. I came home, I started doing, I always did landscaping, and I started doing my own gig, and they called me back and said, hey, you want to come back and work? And so I sure enough did. sounded like flying around a helicopter and float planes was a good time. So I joined up with him for a stint as a seasonal employee. I did about 7 years. I finally got hired on permanent as a biological technician at the Craig Ranger District. And during that time, I was there about, let's see, I did about 7 years as a permanent employee there. I spent about 15 years total in Alaska. I did 2 3-month stints with the Alaska Department of Fish and Game, and I only will touch on that because we'll cover a little bit of it later. Those were 3-month stints, one other person, myself, on an island studying coho salmon, but primarily. But it was a pretty unique time in my life to be able to do that. And there are still some folks doing stuff like that, but it's few and far between to be that remote for that long a time anymore. I then left because I got sick of rain. Living in a rainforest, 100 inches plus a year. I got sick of that and decided I needed to dry out a little. So I moved to Wyoming. Part of the reason we all know is we end up with people in our lives that help us along the way to make decisions. So myself and another person, my gal at the time, we left Alaska, we moved to Wyoming, and I took a job as a state fish passage biologist in Wyoming. And that was a great time there. We'll cover some of that. The Agricultural Research Service portion of my job is still with the USDA. So I'd worked with the Forest Service as a permanent employee. I had reinstatement eligibility with the government. And again, the gal at the time decided to go to grad school in Milwaukee. And when I showed up there, I said, I need a job in the fisheries world. How can I turn this into something? And there happened to be an Agricultural Research Service lab right in Milwaukee, and they were hiring for a perch animal caretaker. And so that was a great introduction into really, truly science as a career, not just field work, biology, and management is really what I've been doing up until that point. Then we moved back to Wyoming for a period of time, and I put ski bum in there because I did do that for a little bit of one winter, but I ended up working for the Forest Service again there. I did some GIS work. I then moved into the administrative world and ended up here in Wisconsin as an administrative services specialist, touching pretty much every piece of paper that came through our lab here and working directly for our project leader, the manager of our facility. And then lo and behold, a job opened up as a biological scientist, and I thought this is a great opportunity to kind of get back into the science, get back into the woods, and you know, do the things that I love again, at least hopefully. I don't know if I can do 3-month stints anymore, but that's where we're at. So Randy called and said, hey, Tim, would you do a talk? And I said, yeah, man, I can talk all day about all kinds of things. What do you want to hear? He said, oh, you talked to my brother. I said, well, I've been on tops of mountains. I've stood next to waterfalls. I've flown all over the place into unique places. You probably can't see it, but right in the center of that photo is a wolf print that's like the size of that GPS unit sitting there. So I've seen some pretty unique animals while I've been out there in the world working. This is a little buffalo I ran into in Wyoming. The bighorns, and even some fishing spiders that we got into last year that were a little bit terrifying to me, but Sarah there didn't mind getting her finger close. And then Randy said, no, Tim, I don't want you to talk about Alaska the whole time or being out. He said, I need to have something that's going to be applicable to these guys. So advancements in science and technology lead to better tools for managing habitats. And I said, that's probably something I can speak to a little bit at least. And then we started talking some more, and he shared what you guys are doing, and it does fit very well with what my past is, some of the things that we're working on now, and really exactly what you guys are working towards. So I thought this is something I can do. So here we go. This is really the main part of the talk, but I'll share my email at the end, and I'm always willing to talk about anything else, but here we go. We're going to look at some advancements in science. And to start that, I've been doing this type of work, let's say, at least being involved in natural resources management and science for about 30 years. And what's changed in that time? And I kind of wanted to speak to what I knew and speak to what I've done, and then also kind of give some indications of the things, you know, just technology in general that's changed. We all know some of these things, but how it's really impacted our work. So we're going to talk today about maps and navigation, the changes that have occurred over time, fish sampling, different techniques and things like that that I've been involved with, some of the things that might, you know, relate directly to what you guys are doing, and then some of the new things that we're working on here. I was the state fish passage biologist, so I've got a pretty good understanding of that, and I'll kind of speak to some of the changes. And really, in this case, my eyes were opened when I realized after leaving Alaska, where we did have our own fish passage problem, so it can be wrong, as to what the lower 48 in my world at the time was dealing with. And then, of course, fish habitat, that's what we're all really keen on if we're in that world, because that's what's going to give us the biggest bang for our buck and keep our fisheries healthy and the fish themselves, you know, moving in a good direction. And then I got genetics and genomics on here. That's not going to be a huge part of my talk, but I am going to cover some of it because that's really my world at this moment. And I'll talk a little bit about what we're doing here in the lab, some of the things that we have going that I think overlap directly with what you guys are doing, and where we're going from there. So let me take a little sip here. I've had a sore throat for the last week, and so if I start to get scratchy, let me know. This is a photo of Brian, former colleague. He's out west still, Oregon. He's a hydrologist, and we're working on a in-stream, catch up to the same slides you guys are on this screen so I can watch it. And we're working on a in-stream wood placement. This is a large river system on the island. And Brian says to me one day, and this photo is taken after this, and we'll talk about that a little bit, but he says, someday we're going to have one piece of equipment that can do everything, and we won't need to carry all this stuff around with us. And at the time, let's see if we get to the next one. At the time, 97, and that was 2003, but we pretty much had this exact stuff. I know somewhere, here we go. We pretty much had this stuff here. We had a map, a contour map, in our case, 100-foot contours, which you remember that photo, that mountain almost looks flat. We had aerial photos, 4-inch to the mile. You can't see virtually anything because the entire area is forested. We had a compass. We had 28 degrees declination. If you don't know what that is and you go there, you'll probably get lost. We had clinometer, which is gradient, right? So we have hills, so we need to know how steep these are. An altimeter, which varied based on the weather, and in a rainforest, a lot. So you never knew exactly how high from sea level you were. One nice thing was we did live right at sea level all the time, so we were able to set those pretty regular based on that. And a VHF radio to communicate, right? And that was our communication. No cell phones. Cell phones existed at the time. My mom had one in Indiana, but no cell towers on the island I was living on. No way to communicate other than VHF radio. And the Forest Service, just to kind of give you a rundown, they have one of the best radio communication systems in the country of anybody, primarily for firefighting, but we used it, of course, for our work too. Float following with boats, helicopters, float planes, that type of thing. and we could talk to each other um, very well, almost as well as we can now in the lower 48 with cell phones. We could in Alaska talk by VHF radio, which was to me impressive in 1997. By 2024, so today or this year, this week even, I should say, Monday, went out, 11-hour day, went out in the woods, do some work. We took with us one iPad. It's got everything on it: camera, compass, GPS, all the software. We got our points. We can connect to the internet. We can email if we want to right out there in the field. That's where we're at today. This is where we were. So I'm going to kind of recoup. There we go. So I'm going to kind of run through my history here. So GPS was a big part, right? I had a field map that had 100-foot contours on it. And maybe 1999, I'm guessing. 1999, so first GPS military. In 1999, I touched the first GPS unit I did ever in my life. It was a U.S. military thing. I had to sign a deal that said I won't turn it over to the Russians or whatever it said. And I thought, man, this is incredible. All it gave us was the lat long. So in 1995, we got full operational capability, and by 2000, see military accuracy available to all users. That means that in the next, whoops, that's not the slide. So I was carrying around in 2000, 2001, a giant backpack. I want to back up one slide. This is about the size of the unit they gave me that was military grade in 1999. And I was carrying around a backpack size. If you guys seen a shocker, I know some of you have, a backpack shocker from Smith Root. That's about the size of the GPS unit I was carrying around. So the military has had a much better handle on GPS and much better handle on the handheld version than we have in the private sector and government, but that's later. So that's kind of the history of GPS. Well, that was great. We can tell where we are on a map, but we need to be able to deal with some of that map info. So I don't know if anybody's ever heard of Esri. Most of us have heard of GIS, probably, I'm guessing. And nowadays, almost everybody is using it, whether we want to or not, because we got to figure out how we got to get to our neighbor's house. We can't even read a paper map anymore. I just tell Siri, I want to go to my neighbor's, and they direct me there without even opening it. So I want to point out a couple of things here with GIS. This is Esri, the company that really has kind of led at least the government's GIS world. 1982. This is their first thing. This is back when most of us were looking at, if we were around then, green screens on and cursors and that type of thing. A mouse wasn't even heard of. And I want to point this out right here: tightly scripted software code. Okay, so that's how I first learned to do this work. I learned how to do GIS work using code. Then in 1999, we got ArcGIS. ArcGIS, we had Arc in 92. So I first used ArcView, which is a map viewer. ArcGIS allows me to input my data and relate it, because this is a relational software, relate it to other data. It doesn't just allow me to put it in there and put it on a map and make a map. It allows me to say, this stream is here, and this type of vegetation is here. Now I can relate what other streams have that type of vegetation and do that across the landscape. That was a real powerful tool that came around in 1999. Early in my career, mind you. I want to kind of fast forward. This timeline ends. I got this off of Esri's website. But in 2024, we have gone from point and click in desktop, Windows type thing. We use our mouse. Now we are back to, and almost all the GIS, heavy GIS users I know, are back to using script. It's just more powerful. It's quicker. They can build their maps that way. It's not point and click. So for the lay person out there, GIS is still available. We got it on our phones, right? We can map ourselves to places and even collect points. But for real heavy relational reasons we're using scripting now. So that's one thing I just wanted to kind of highlight. So to get back to Brian's thing, we got the start of something great. Apple iPhone comes out in 1999, right? They don't even have an iPhone until down here 2007, but they had the idea in 1999, right before Brian said we're going to have something all of us can use. In 2003, remember, GPS is now out. Google's finding that 25% of its searches, Google didn't start that much earlier than that. 25% of its searches were of geospatial nature. People are looking for how to get to grandma's house. That's what they're doing. So Google says, hey, we better do it. And they create Google Earth. That is the start of the world that we live in now, where we can connect with the map out there. We got the iPhone, Brian's wish in 2007, and then this is the day he's been looking to for 2.5 years. So Steve Jobs, Google, and the likes of them, those two companies have really kind of been at the forefront for those of us that are not in the military and bringing some of that. So what does this mean for us as fish people? Let's call us biologists, people that care about fish. We do some work, and so one of the things we need to know is where are fish, right? So we have some techniques that have existed for. When I first started, we were shocking fish to figure out where is the end of fish habitat. That was really one of the reasons we were doing that, is because we were doing a lot of harvesting, timber harvesting where I was, and there was a buffer on fish habitat. And so we needed by law to say, if there's fish here or it's fish habitat, we want to make sure that we are protecting that, because by law, not everywhere has that either. Not everywhere has a law that says this buffer. Some places, some forests at least, have only a regulation that says that. So there's some differences there. Also, snorkeling was a big one for us. We could go through and count fish or see fish and count them, and then hook and line. And I put some photos in here, and some of you all might recognize some of these people. I needed a good photo of shocking, and I just went through your webpage, and this one seemed to me to be a great use of that photo. So I'm glad to hear you all have been getting involved with that, and isn't that a great technique to see fish? Here's my buddy Jeff. This is where we're working on a project, a steelhead project, and he's got his hook and line and his wetsuit, dry suit really, for doing his snorkel sampling for steelhead all at the same time. What a guy. So some of the things that we've got, with some of these techniques, we've got great positives for handling fish with these, right? We can get physical fish data, measurements of the fish. We can handle the fish, look at the health of the animal. And we can, because we're handling those fish, we can do some types of studies. I just highlighted recapture studies, but there's others that we're doing. So we can catch a fish, mark it in some way, and then we can release that fish back and see how many times we catch it or how often we might recatch it, and gather some information about the population. We'll get into that a little bit later. So there's also some negatives. These types of sampling can be hard on the animals. It's not always, but I got a little photo down here in the bottom. And this may or may not be, but it's very similar to some I've seen, where a shocking in a small fry, and I think this is I believe that is a young of the year coho. It's kind of hard to tell with the glare, but and this is what would happen. Occasionally, not all the time, with shocking, you can damage a fish. It just tenses up their muscles. It kind of contorts their body, and it can be, and this fish is healthy. He's swimming around doing his thing, but this can be hard. They also have a high cost versus a reward from time to time, because we can't sample every single square inch of a river at once. And so part of that means we either got to have a lot of people and we got to have a lot of money to, and then we got to have a lot of equipment to cover an area real well using some of these techniques. So we're always looking for better techniques to do that. And so we'll get into some of that in a little bit. Let me catch up myself here. So the other thing we're doing, and it sounds like you guys are doing too, is habitat access, utilization, some of the things that are going on along the shore, along in the river, large woody debris, things like that. And so sampling for habitat access or utilization of that habitat can have some of the same effects. And I'm going to talk about a couple studies here. We've in Alaska, there was a big push in the 80s and early 90s to install fish ladders. Fish were not able, at least the pink salmon, this would have been totally feasible for a coho, but pink salmon in this case, were unable to access upstream of this. And there was many miles of great spawning habitat upstream of this. So they installed, oh, I'm over here, let me point this out. They installed these fish ladders, many of them across Alaska. It's great. We would go up and count them, but the thing is, it's hard on the fish to get trapped in this little box up here with Jeff standing. And there's only so many will fit in the box, so fish are coming up here and then they're swimming back down and swimming back down. And so that can be hard on the fish. This is, and we're going to look at this area in greater detail, but this is a net I've got stretched. And this right here is a channel on a river. This is actually the outlet of a fish pass. So here I am netting fish to see what's using this fish pass. This is in Wyoming, by the way. And we'll talk a little bit more about what's going on here. I do want to point out right here, this is a diversion. And that is, we'll talk about as we talk about fish passage here, what that means. And then you guys were talking about pit tags, and I took this off of California's because I didn't have any photos, but pit tags, you know, this is harmful to the fish, at least to some extent. However, great set of data that can come out of this stuff. So in our sampling over my career, we've had some of these techniques come along that we are using and still are using that provide us great data, but can be, I'm back up a slide. Oops. They can be hard on the animals, they have a high cost, and the logistics can be challenging. Okie doke. So another thing we do in the fish world and in any natural resource or at least species world is we're trying to often obtain population estimates. And so these require a lot of time, effort, personnel, maybe not a lot. Like I talked about that Chuck Weir project, 2 of us out there for 3 months, not a lot of people, but it was a lot of time and a lot of effort. They're challenging to implement system-wide, and I can explain that in a little better detail in a second. And the impacts on the natural movement are often extreme. And they require some harm to the animal from time to time. So we talked about the mark recapture. That's one way we can come up with a population estimate. The other way is, and this is a weir. I hope everyone maybe has heard that term. Basically, this is looking upstream. This is a bidirectional weir. This person's looking in where the fish are captured. This totally covers from one end of the channel to the other, or one side of the channel to the other, I'm sorry. And allows, there's a snorkeler in here checking the weir for, we're blocking all passage. They must go in an opening here, and they must come out, in this case, an opening which would be over here, but we haven't yet installed it. This is very early on when we have just installed it. Photo. So every fish that's going to make it up our river, this is right at saltwater, or less than a quarter mile from saltwater influence anyway. Every one of these fish have to come in this baby, go upstream from there. We gotta handle the fish, take our scale samples and all the things we would collect to gather that estimate. And then we would release them upstream. And then we would snorkel and count how many of these fish, and I didn't put a photo, I can't put a photo of everything, but we would mark these with floy tags. I talked to Randy, he said, it sounded like you guys had done some of that. So we would snorkel that whole river, at least the stretch of river from a waterfall where they had no more access, all the way down, counting fish that had a floy tag, and then counting fish that didn't. And we could come up with a pretty darn good estimate of how many steelhead do we have in this river by doing this. But that's a lot of time, a lot of effort to get this thing installed. I put this in here because this is where I lived for 3 months on that Chuck Weir project. And this baby right here is about a $15,000 piece of equipment. And what it does is it puts the head of a salmon like this, goes right here. We got two different sizes for small and large. And it puts a coated wire tag that's individually tagged into every single fish that comes down. We captured 98, 97, 98%, I think, is most of the time as the fish were leaving to go to saltwater. That means every fish that comes back has a little tag in their head, and we can tell a lot of information by an intensive study like that. The problem is it takes a lot of time, it takes a lot of effort, and you got to wait 4 years for some things to come back. And then we got to harm the animal to get that information, to get the follow-up. So those are a couple, great studies that came out of that. Chuck Weir, Chuck Creek is an island on Hecata, and they use this was for the fishing game in Alaska, ADF&G, who I was working for as a short-term assignment, and they use that for commercial salmon estimates on small streams, and that they would extrapolate out to the quantity of streams that they knew produced fish, and could use that to sort of hopefully measure how the fishery was going to do over a period of time, and then predict what they would allow the catch to be. So the other thing we talked about is sampling for habitat. And, the main thing with habitat is we're always looking for what's the proper functioning condition. We don't want to make, let's see, what would be a good one for you guys? Well, we don't want to make barracuda. How about that? That's an easy one. We don't want to make barracuda habitat in a cold water stream. So we need to check out what the proper functioning condition of this is, which means we need to cover a lot of ground. Takes a lot of time. And covering that ground can be pretty logistically challenging. And I put some photos in here. This is me. We're actually capturing. This is Brian with his fancy dancy. See, this is after 2003 when we first met. This is probably close to 2009, 10. He's got a total station here, laser total station in this rod, and we're totally mapping the channel of in I think 1,000-meter reach is we were able to map very detailed as they did in-stream wood structure placements. These are obviously holding some of these root wads and things down, but we were able to map and then see the change over time to try and reach this proper functioning condition. The problem with habitat is there's a lot of information that you want to collect, but some of it is hard to repeat or get you and I, let's say, if we go and look. If I come out here, I'm looking at a big, and I don't have a good one here, but a big wad of logs, and I want to say how long those are, and I need to say how wide they are, but the water's moving fast. I got to estimate that. And then somebody else comes back a year later, they got to estimate it. Those numbers don't always jive, and they might say there's more when there's less, or I might say there's more when there's actually less. And so some of these, we still use data sheets like this. This was one from a study we did, or we're working on even still currently. Some of these data are really easy. Velocity, we got a meter, it'll tell us exactly the measure we need. But this large woody debris, right? We're putting it in 3 size classes, but some of those you can't see. It's murky water. I don't know how far it goes, and it's too deep for me to touch. So some of this is estimated, and that therein lies the problem is some of this stuff becomes not repeatable. And so that causes problems. Not problems, but it just makes it a little more challenging to really say a lot about it, to get the right answer out all the time. So these next few slides are going to be some, they're kind of I didn't really put a whole lot of text in here, but I'm going to talk about some stuff. So I said I was a state fish passage biologist in Wyoming, but I had done fish passage work in Alaska. In fact, the first project I ever worked on with a GPS was what we termed at the time road condition surveys. We called it RCS. The government's great with acronyms. So we had RCS. They gave us a GPS, and we went around to every one of these culverts, every one of these bridges, and anything to do on the road, gates and all this stuff, and took a point and said, here's what is here. This culvert lies here. And as part of that team, there was someone like myself, a fish biologist and an engineer. And we did that specifically, and that project started in 1997, I believe, was the first year they started collecting data. Part of the reason we did that is have a fish biologist and an engineer is because the engineers really know the road part of it. But the fish people can tell the fish habitat and the stream characteristics. And so what we did is we went around and looked at all these structures. And some of them look like this, where a fish will never make it up here. You can see this vegetation. Even at the highest flows, this baby's only half full, maybe a little bit more, but it's shooting a torrent out of here, and a fish will never be able to jump up in there without some substrate. So I had a pretty good understanding of fish passage. And of course, we talked about this is looking down at that fish passage that I had showed in that earlier slide. So I knew we had fish passage. We're trying to, in Alaska, get them upstream, get fish upstream. There's habitat up there and they can't get up here. What are we doing? So we start trying to fix culverts, right? And we were doing that. We were doing a great job of that. I worked on a model, at least for the area I was working on, to prioritize. We did habitat surveys to say the distance and the quality of habitat upstream. And then we started to prioritize. And of course, you can't fix everything at once. You guys know that. It takes money and time. But we really made a pretty concerted effort on the Tongass in the early 2000s to begin that process to allow fish to access habitat that they've been unable to access for many, many years, sometimes decades even. So then I moved to Wyoming, and I ended up as the state fish passage biologist. And I want to point this out because fish passage is a big thing, especially for trout and some of these higher gradient fish that'll use these higher gradient streams that are a little steeper. In Wyoming, excuse me, they have all kinds of high gradient stuff. They've got mountains there, but they got a lot of farmland too, and they irrigate that farmland. And the reason I wanted to show this photo is, as the state fish passage biologist, mind you, I've been working on culverts. I've been working on trying to provide access upstream, and I get to Wyoming and I get out to sites like this. Can everybody see the laser pointer, by the way, before I start talking like nobody can? This is actually
Yes.
an irrigation ditch. Okay. This water right here is a river or a stream. This is a waterfall. This, I'll show you in a second. Right here's that irrigation diversion I was telling you about. And this is the other sidewall with a gauge for this irrigation ditch. So we've got a river flowing over an irrigation ditch in this case that has a waterfall that's impeding fish habitat. And I'm sitting here going, man, I tried to get fish up culverts, no problem. But here I'm trying to get a fish up a waterfall. Not only that, I've got fish in because they're diverting. We'll get to a slide in a second. We'll show you some of these diversions. They're taking, in some cases, 90 or 100% of the water and forcing it into an irrigation channel that's going to get dumped on a field. And all the fish in the river are swimming right down into the irrigation ditch and out onto the field. So we started trying to figure out, man, how are we going to handle some of this? So the first thing we needed to do was to start to put together. It was me and my boss in the fish passage program in Wyoming. And really, I would say, and he started the program. He did a fantastic job. It's gotten picked up by several other people over the time now. That's been about maybe not quite 10 years since I've been there. And yeah, probably 8 years. And so he did a fantastic job of getting that started, getting these places on the map, and then my job was to go out and survey them. I did this. This is another photo. There's nothing to say here other than this is what our fish passage here. I took that photo standing right here. This is the irrigation ditch. This is the waterfall. No fish can make, I mean, there's an occasional trout that could jump that, but it's very shallow down here, so not a big jump pool. We put this in, and that was part of that other, I showed you that slide that had the spike net or net at the top of this baby. And we had fish moving up this, and it was great to see. So that took some effort, some money, and things like that to happen. But we started a program there. He started a program, and I helped him in the first couple of years of infancy there, and then it's taken over by other people, and they're doing great things out there. I wanted to highlight this photo or put it in just for, this is basically another 2 inches or so, and 100% of this river is cut off, and all of it will be headed down through this irrigation diversion, and that's what I was talking about. So when I got to Wyoming, I was thinking, man, fish passage, this is going to be awesome. Working in the woods, checking out culverts, going around. Nope, I'm out in the middle of the desert looking at these irrigation diversions on these rivers. But it was very exciting, very interesting work. And I got to talk to a lot of landowners and turn a lot of people's opinions. So that kind of gives a little bit of a rundown until my most recent job work here. And so I wanted to stop at this point, and we've got, so what's changed and what can we do better, right? That's really where we're headed with this talk. And I wanted to highlight this or put this in. This is where we started back in my early days. We had identification books. We had all this old information. We had all kinds of manuals on how to do things and this and that, all paper. By now we've got GPSs that I can put aerial photos on. I can do anything. I mean, I can use that thing as well as I can use a computer. And I wanted to highlight this picture here and really this outline, because what this is showing is here's somebody who's going to go out. I've got a map in my hand, and they're like, hey, I was like, you want a map? No, I'll just take a photo of that map and take it with me. That's where we are today, right? And I wanted to just throw this out because I think you guys are doing a great job, it sounds like, and others are. The education of the public is really what is important to me also, is enlightening folks that some of these things that you guys are doing really do make a difference. They make a difference where you're working, but the same thing can be implemented all over the country. and is being implemented all over the country in some ways. Okay, I am going to pull this baby up real quick and run down another one of these so that you can, and I'm going to, can you see this one, USDA? Yep, good. So,
Yes.
so the Forest Service, I work for the Forest Service now and have for many years, but we all fall under the USDA. I'm not going to go into this, but I wanted to highlight a few things. The NRCS is instrumental in under USDA in providing private landowners and entities with money to improve habitat and do things, and they're a great resource for especially private landholders in improving habitat for fisheries, for erosion control, for many other things that can benefit fish and other wildlife species. Here's where we got a Undersecretary for Research, Education, and Economics, and we got an Undersecretary for Natural Resource and Environment. The Forest Service falls under this Undersecretary. ARS, the other research branch of the USDA, all of the power of ARS falls under a different group. So I just kind of wanted to highlight that where I work now does not fall under the main arm of the USDA's research. So we got the Forest Service, comes about 1891, Transfer Act of 1905, takes it out of Department of Interior, which we still get our money from, the Interior Appropriation, and it creates the Forest Service, 1905. Gifford Pinchot, first guy under President Roosevelt, and I just had a discussion today. Gifford Pinchot's grandfather works for the Northern Research Station. She's a geneticist, and she's working on one of the, it's either emerald ash borer or there's another chestnut maybe that she's working on, but it was very interesting to me the first time I heard her name. I said, oh, she's got to be related. You don't hear that name very often, so I just want to point that out. Under management, so I work under the research group, but here's the management: 193 million acres in the U.S. We get a $5 billion budget, and that's a half of it spent there, and we get about, I don't know, the research is minimal of that. I'm going to zip through a couple of these. What we don't do, we're not the Interior and all of these groups. We're not the state and all of these, and I could throw Massachusetts in that. And we don't manage species. That's what's funny. We manage the habitat generally. There's a couple caveats to that, and I'm not going to go into it. But suffice to say, Alaska's got their own deal because the state constitution prevents them from following the laws of the country. And that's a much larger discussion to have. So they do manage some species in Alaska, and I'm sure other places. So we're a multi-use agency. We also have wilderness and national monuments. We do a lot of fighting fires. That's what we're here. So we were the Forest Service broken into regions. Region 9 will be you guys. I want to point out that Region 9, let me put this on again so you can see me. Region 9 is from Maine to Minnesota, right? From Arkansas, the border, all the way over here to basically West Virginia. And all this is one region. That's the Northern Research Station boundary as well, which you'll see in the map here. This is our research stations. This is the Northern Research Station's boundary. That's who I work for, the Northern Research Station. So we cover an area quite substantial in the country. We have scientists in these locations here. We got 14 research units across this area of our Northern Research Station, one of which is right there in your neck of the woods in Amherst. We've got a great guy, and I told Randy about him. Keith Nislow is his name, and he's a fisheries biologist, fisheries biologist right there in Amherst. So he'd be probably a very great contact for you guys. At the Northern Research Station, we've got some focus areas, and I'm going to touch on a handful of people. But in my lab here, we have 6 or 7 scientists now. We lose them occasionally. One retired not long ago. I'm trying to still add up in my head, and then they merged some people in. So I think we have 7 right now. But we have people doing modeling of forest disturbance, computer-based modeling. So they're basically in a computer simulation, growing forest and then introducing disturbance into that forest, representing the natural environment. And they're introducing fire, they're introducing insects, introducing wind, and a multitude of other disturbances that they're looking at, including pathogens, by the way. We've got natural resource stewardship, and I want to highlight one of our scientists here. They do phytoremediation. That word is a big word, but it means basically they're taking the crud out of dirt. So they've got a tree we developed here at this lab, a fast-growing poplar tree, and there's a couple other researchers around the country who are involved in that suck up water faster than most trees, just because that's how they were built. And we didn't make them, we traditionally bred them, but that's how they do it. And he's using that to go to old landfills and is studying what of these trees can we find that will suck up and do good water quality, suck up the crud, heavy metals, and some of these other problem things coming out of landfills. Sustaining forests, of course, we have a forester, a silviculturist. We've got a plant physiologist. We've got water and air. We're part of that group also. And then the big one here, inventory monitoring and assessment. One of the largest, at least people-wise, entities in Northern Research, in all the research stations for the government or for the Forest Service, is the FIA, Forest Inventory Analysis. And they are doing a long-term, and have been and will continue to do, long-term assessment of changes in forest over time. And then that is feeding into some of these models that we're using and developing here. That's also helping with landowners who are wanting to grow better, and mills and things like that, know what's coming down the line. So some of the widgets we produce here, just to highlight, we've got publications, all the scientists here. I should also state, I am a side scientist. I'm not a technical research scientist. I don't have to publish. I just get to help other people do their publications. But we have scientists here who have been instrumental in developing software techniques, models, other things that are being used by other researchers around the country. And one other thing that we really hope to have is, we have some here, other places do, especially our Forest Products Lab, patents. If we hold a patent, that means anybody can use it. If a private company holds a patent, then they can license that out and make a lot of money off of it. So I don't ever say that the government should be in the business of seeking patents, but I do think sometimes it's important for that to be public information and public use. So and the other thing we do is experimental forests. And I just wanted to point out one, that this Hubbard Brook is right there. It's in your neck of the woods. It's not right there, but this has been an ongoing, and I don't really know that much about it, but I read some about it. It's an ongoing, long-term study on stream flow. I'm sure there's a fisheries component to that, and other things, the habitat associated with that. And so this is something that probably Keith would know a lot about, and others in that area. And feel free to look up our website. We'll take a look at some of that. The one other thing we do is research natural areas, and these are around the country, and I won't go into it, but There's 51 of them in the north, in our region. Okay, let's get out of this baby if we can and go back to my other one. Don't know what we just did. And perfect. Okay, so here we are again. What's changed and what can we do better? I want to say, in my experience, one of the best things that I have witnessed is the growth of groups such as yourself and many others who are promoting what they care about and helping and partnering and collaborating with other entities, including universities, state, local, and federal government agencies, and other entities. And we also have, Smith Root is, of course, a public company. Waste Management here is a public company. We have a scientist here, the phytoremediation group actually has a partnership with Waste Management and is doing a considerable amount of work with and through them to help remediate some of the problem landfills that they've taken on as they acquire some of these other companies. But let's get to the fish side of things. So aquatic species sampling, we talked about that before, and we were doing it with shockers, we were doing it with nets and fishing and that type of thing. And so today what we have is this is me here, this is my colleague Rachel, and this is our his name is Chris. He is our counterpart on the management side. So he works on the Hiawatha National Forest. He's the fish program manager there, and he lives in the UP. So we're in the UP here. And at this point, what we have here, this is Sarah. This is a study that we're doing for lake sturgeon. Attached to this thing, you think it looks like a shocker if any of you guys have been out shocking. This little contraption here, and this is the end of it, and Rachel's wearing gloves. This sucks up water. It has a tiny little filter, a disc filter. It's about the size around of a golf ball. And we filter water through that. Whatever collects on that is coming from a living organism is trapped it's a 5-micron filter, so it traps almost anything. I mean, but the water itself, and even that it slows down. And whatever's trapped on that, we can extract the DNA out of. So what we're doing is we're going out and saying, hey, we want to know what's in this stretch of river. We go out, we take our filter, we attach it to our rod, we stick it in the water, we run it through our filter, and then we bring it back to the lab, extract the DNA, then we got a vial of DNA, and we can then take a look with a very small amount of that and test for species of concern to us or whomever might be interested. So we are just getting started at our lab with this. There's another lab out of Missoula. They've been doing it for quite a while, and we are partnered up with them on some projects, and we'll talk about a few of those. One of which is a trout project. We actually have a trout project which is involving brook trout, cutthroat trout, and brown and rainbow trout. And one of the things that interests me, and we, some of us might talk more offline, because it sounds like you guys are collecting some genetic data yourselves with maybe the USGS or one of the Massachusetts groups there. I didn't write it down, but we might be able to share some resources there. I feel like that and see, because one of the things that's going on is out west, they've got brook trout that are not native. They don't want them. You guys are trying to get more brook trout because they are native. And so one of the things that, and Rachel here in this photo, she's a geneticist. But really her specialty is genomics and bioinformatics. And so what she looks at is the differences between organisms and their populations. And so that's a very new field. I'd say genomics, maybe 20 years. The term has been around, but what it's taking, and I don't think I've got a slide that says this, but it takes very high through, very large amounts of data. So we got to sequence the data. We didn't have that technology until we probably had it 25 years ago starting, but the more recent and very good quality, high throughput stuff is much more recent. And we didn't have the processing power to handle all of the DNA sequences that we would produce. And then I put this other piece in here because in this photo with Rachel, we're actually out. Hang on just a second. We here are collecting. We're out trying to see. We're at habitat for dragonflies. In the photo down here with Sarah, this is a stretch of river. We're trying to determine if we have lake sturgeon. In this case right here, we're doing the old school way. We've netted in this bog, basically, a bunch of insects up because we're looking for the adults. And the reason we need some of the adults, the dragonflies we're looking for, Heinz emerald, are endangered. But there's a group out of South Dakota that we're part of, University of South Dakota, and they're looking to collect some of these because we need that to get our actual DNA that we're gonna, if we say we have DNA, we need to test and make sure our chemicals and our chemistry is right, that we are saying we have it and we do have it, and we're saying we don't have it if we don't have it. And so we needed a handful of adults, and so they had a permit that allowed them to collect some of that stuff. And so I put that in there because it still takes a little bit of harm to an animal. Even some of these techniques that we're using now that are far less invasive, obviously. So here's the setup that we're working with now. We're into a lab. This is all the tubes that it takes me. Each one of these tubes, there's 24 of them. That's one run I can make. That's about a day's effort to process through 24 of those filters. And it takes 1, 2, 3, 4, 5 at least, and there's probably one missing, to tell you the truth. 6 at least sets of tubes. Yeah, right there it is. 1, 2, 3, 4, 5, 6. Yep. 6 sets of tubes before I can get to where I have my DNA in my hand. This piece of equipment right here we just acquired. It is an auto pipetter. Like I said, we're trying to use very little quantity of this DNA for a couple reasons. One is we'd like to be able to use it to test for other things in the future. If we went and said, hey, we got lake sturgeon here, we may want to know, do we have red-side dace? Do we have the dragonfly in the areas we have lake sturgeon? We doubt it, but it's possible. So we don't want to use up all our DNA at once. So we bought this auto pipetter, which allows us with 10 microliters, which is about three teardrops maybe or less worth of material, worth of liquid. It can handle that type of that very small volumes, and we put them in these little trays, and then we run them through a qPCR cycler, and it'll give us a reading that says yes, you have lake sturgeon DNA, or no, you don't. The process is it's amplifying that right up. Is it really 7? I better go. No, here we go. We got some technology to make fish passage better too. I showed you those other things. One thing we're doing is much better structures these days. We've got engineering and things that are, if we lose a pipe or we got to replace it, we're figuring out how to make this stream really much more beneficial. It has substrate in the bottom, allows the flows, doesn't get blown out. But these are some interesting things that we had in Wyoming. This is a moving screen right here. Water flows through the screen, it self-cleans itself, and so it doesn't clog up, and it runs water out through an irrigation ditch. These are the same thing, they're just drums. And this one, the water flows in here. This is a screen. Eventually it will fill up and it will spill over, but these screens can be cleaned. And those are some techniques that were developed, to say, hey, we want to keep fish out of these irrigation channels. The one problem we got is challenges that still exist in gathering our habitat data and things like that. And so, one of the things is access. It takes you've got to cover some ground, and a lot of that stuff is remote. You can't get some of this information, for instance, velocity, without having some equipment out there in the place. And then I put bugs because, man, I hate those. And this is a wealth I got. And kudos to this gal right here. She's a trooper. All these men, bug shirts. She's out here doing the work while they stand around without a bug net. She's from the UP, and the women there are incredible. They're used to that stuff, and I don't know how they do it. So here we go. This is some of the stuff that I'm gonna go back. This is really where we're at in our lab right now. Rachel's doing some other things with models, and I won't go into that. I know we're running short on time, but this is where we are with the Forest Service. And let's get back to I think I can get back to nope. Here we go. Technologies, new ways to collect habitat data, and the Forest Service is capitalizing. So I'm going to open up a couple of these. Maybe I've got them open and I'll just drag them over. That's probably easier. Whoops, wrong thing. Here we go. I just wanted to highlight, and I don't know if you can read that, but let me zoom in a little. Control that. Oops. Get over there. So right here, this is a main channel Au Sable River in Michigan, but they're using drones to fly these rivers now and highlighting. This is warm water. This is cool water. Right? Red color is warmer than ambient winter. So this is cold water in the winter, warm water in the winter. And in the summer, this is warm water and cool water. And so that's one of the techniques that's going on right now is some of these things that have come around is drones. And the technology is there now to give us some of these benefits. Mind you, these are right out of the Forest Service research. And this is 2003. This one is ongoing still. Here's the folks. But you can find these right on our website, Northern Research Station. This one's out in Oregon. So it's not a Northern Research Station, but same principle. They've got these toads. I don't know. I thought there was maybe it's in here. But this is a giant 2,000-acre bog, and they're going around looking with drones, flying this whole area in basically a day, in order to tell what's the water temperature of these areas, and where can we prioritize to go and look for where we might find these endangered or at least threatened or imperiled spotted frogs and other species they might be looking for. And there's one more, and I don't know why it didn't open. Let's see if it does. I don't know why that's, well, we're not going to get it. I'll just highlight this. So drone again, in this case, this is forest structure. This is done by LiDAR, which is a technique. We're now bringing LiDAR into GIS and using that. We were using LiDAR for elevation and showing basically the equivalent of contours. But somebody figured out that, hey, man, if we crunch this the right way, we can actually start to see what the forest looks like. We can tell where it's very dense in in the understory and short maybe. We can see where it doesn't have much of an understory. We can even see the tree trunks. Oh, you probably can't see that. We can even see the tree trunks, and we are going to be able to get a height and elevation of this. We can calculate biomass right out of some of this stuff. So I told you about some of our guys here doing this modeling, forest succession modeling. Brian and Brian Sturdivant and Eric Gustafson. Eric Gustafson just getting ready to retire, super scientist. And by that, I mean, that's a technical term in the government. He is at the top of his game, and there is only, and I don't know the number, but very, very few super scientists that are in the government system. One has to leave before they allow one in. He was instrumental in developing this landscape disturbance model. And as part of those models require good data inputs. So they're now starting to incorporate LiDAR data to feed these models better information, in real time. I don't mean real time as while the machine's flying, it's updating their model, but they're going out and changing, saying, hey, this is an oak stand. What does this look like? What's the biomass? What's the diameter of these trees? What's the understory? And being able to then feed that information into their models and better produce a result that will realistically depict what's on the ground and what can happen. So these are some of the things that the Forest Service is using. I'm excited to be back in the science world, working with the genetics here, working with Rachel. I have not spent a ton of time in a lab in many, many years, and it's been interesting. And so now we're out, we're collecting these filters, sampling for fish, and then we're going to the habitat and fish habitat managers and also the fish species managers and saying, yes, you do have fish. Here's where you have them in high numbers. Why are they there in high numbers? And then we're using that information. Another one of our scientists is putting together habitat suitability models so that these managers can say, hey, we now know where they're using, maybe for spawning, maybe for rearing young over time. These are areas that are important for us, but what do they look like? We're collecting habitat data on that and saying, okay, if we want to increase the number of lake sturgeon, let's say, or increase the number of, in your case, trout, what does it look like that they are using and where is that lacking? And how are we going to best be able to promote that? And it sounds like you guys are doing that. I don't think that's anything new that we haven't been doing in the past, but some of these tools are better able to provide us without damaging the fish and the drones especially to provide us habitat information. Drone flights are cheap, and as opposed to sending a guy up a river, walking, collecting this water temperature data, we can go out once a week and for 52 weeks have a really good idea of what the rivers are doing over time. And we're just park a truck, fly it up a river, and bring it back, and away we go. So anyway, that's kind of the talk I have put together. I hope that's I didn't run too long there. Randy didn't yell at me yet, so and I will gladly take questions, or if everybody says, hey, we're done with this guy, then feel free to let me go and we'll chat again later. That sounds fine. Thank you for letting me do this too, by the way. I did not know what to expect, and it sounds like you guys have a very good understanding of what it takes for fish to be productive in a river, as well as have some great contacts out there doing excellent research and on the ground restoration.
Thanks a lot, Tim. Enormous amount of information and some exciting stuff that we definitely want to look into, and we'll be talking with you just to try to get good leads for pursuing some of this stuff or looking further into it. does anybody have any questions at this point?
Hey, Tim, thank you again. I just texted your brother and told him you were rocking the stage like Bruce Springsteen here. So
Yeah, I took my glasses off. I don't know if anybody knew. My glasses are broken. I don't wear them very often, but I thought for this thing I better at least be able to see some faces. And
we
thanks
didn't
for
know
nodding
until you,
and that. So
Tim, we didn't know until you told us. So,
yeah,
you know, we couldn't tell.
I've lived in the woods long enough to know I don't care about what people think of how I look. So
Do you have a
yeah, one
question,
of these days it would
Andy?
be fun to get out to see what you guys are doing there. I haven't been to the East Coast in quite a while, but I do have some relatives not too far away there that I plan to visit. So if I do make it that way, I will certainly look you up and try and at least get out and maybe even fish that river you guys are involved with.
We'd love to have you out here. I just have a real quick question that
Sure.
probably a yes or no answer, actually. It sounds like the environmental DNA studying work that you're doing, you're able to get quantity, like you're able to estimate quantity of organisms based on that, or just the presence?
Good question. So I didn't have enough time. I didn't feel like to get into some of the nitty gritty of that, but that's a great question. And I did really want to kind of highlight that there are people who are probably going to spend their entire career working on that exact question. Can we estimate a population from a sample like that? The answer is today, no, we can't. Um, we can say if I went up a river, and let's say, and we did this on 3 rivers, in fact, for our lake sturgeon study. Every 1,500 meters, we would take another sample, or about that distance, wherever we had good access and public land and that type of thing. And so we can quantify. We have a quantity of DNA that we captured on our filters, but we can't relate that to a number of individuals. So estimating population using that technique is not possible yet. I do think that, and from what I've read, there are some. In fact, I'm going to use this thing, but while I'm thinking here, um, there are. Let's just stop share. There we go. There are people who are looking at what does it mean if I have a given quantity of data or of DNA. If I have good numbers of how many fish I have, and I have good numbers of DNA that I captured, then there is probably some relation there. Is that going to be feasible to translate into every single stream out there? If I got whatever, we don't measure in parts per million, but let's say I got 1,000 parts per million DNA of trout on my filter here, and I know I got 1,000 fish in here. There's a 1,000 to 1,000 relationship, so I go to the next trout stream and I get 50. That means there's only 50 trout. Probably you can't do that, right? Nothing that simple. But there probably will be, and I expect initially that type of thing will come from individual streams, is what they'll do. It'll be like in this stream, we know at this given time, if we collect this information, we can then start to say some things, population wise. It probably will not be, right to the nitty gritty. We won't say, oh, we got 313,212. We're going to say we've got, between 100,000 and 500,000 or something like that. Or between 1 and 50 or whatever. But there are folks working on that. And there's another technique, and I didn't get into it, and it's a chip And their equipment's super expensive, so not very many places even have it, even government labs. It's used in other cases, so it's primarily used in the medical field, but it's I want to call it digital qPCR, but it can, so they could take a sample, I believe, and I've only done a little bit of looking into this stuff, but I think they could take a sample of blood, put it in there, and then it's got however many cells that filters into, and then it does the work, and it quantifies the individual amount in each cell, and then that will tell you the quantity of, let's say, cancer cells that are within a given amount of blood, and then they can relate that to some other type of thing. So I think there's folks working on that from a variety of other reasons. When we get that type of technology, or how soon that occurs for things like what we're talking about, I don't hold my breath, yet, let's say.
Yeah.
But I know that's something folks are working on, and would love to have. I mean, that would be the cat's meow really for us as managers, or that type of thing, to be able to stick a thing in, suck up water, and say you got 482 trout in that mile of river.
Right.
Go have
Yeah.
fun and catch them.
Yeah.
So,
Very good. All right. Anybody else have a question before we let Tim go? Thanks a lot, Tim. That was
yeah, you bet. No problem. I
yeah.
didn't pass my email along, but I will. Well, Randy's got it.
Randy's
So
got it. So
share that to everybody, Randy.
Yeah.
And feel free to give him my cell number too. I don't mind that at all. It's really a joy to talk to folks like you guys. For me, that's anytime I can share what I know, which is not a lot. There's plenty of people who know a lot more than me. But get other folks excited. I'd love to just talk to, I mean, I love talking about Alaska. Anybody wants to go, let me know. I'll go along. But yeah, I miss Alaska a lot, but I'm really excited to be where I'm at and doing the things I'm doing, and it's a lot of fun. And I really appreciate you guys, letting Randy connect me with you and share what I do know and stuff that I've been involved with. So
sounds like you had a great career with a lot of interesting experiences, and we appreciate you sharing the ideas with us. And like I said, we'll be in touch to try to follow up. And
yeah, great.
And yeah, very good. Thanks a lot.
Yeah,
Thanks again.
Eric. Thanks, everybody.
Thanks again, Tim. Thanks again,
Have a
Tim.
great evening. You bet.
Have a good night, everybody.